Everyone, I'm here by the names of Adina Miretka from Biomedical Laboratory Science, Devosli. I'm going to present about assignment of applied immunology, and here are the topics which we are going to discuss. The first question is what's flow cytometry and how it is used in immunology. The second question is the principle behind agglutination reactions. How does latex agglutination work in diagnostics? So the question is, what is the principle of precipitation reactions in immunology? And the last question is, how is the radio immunodiffusion test performed? Okay, let's begin from the first question. Flow cytometry is a lab test that uses lasers to analyze the physical and chemical characteristics of cells and particles. It uses lasers, which are light lasers or beams of light, to analyze physically where it should detect, analyze, and count the cells in the chemical characteristics of the cells and particles in general. It's a powerful tool in immunology where it's used to study the immune system and its response to disease. Here it's how it's used in immunology or where it's applied. Firstly, it's applied in immunophenotyping, where they characterizes cell population using antigens on the cell surface and inside the cell. Measuring intracellular cytokine production, where the measure amount of cytokine produced inside the cell. As you know that cytokine is a protein in immune system. So here they applied to measure the amount of cytokines produced inside the cell, as well as white blood cells, which are lymphocytes, T lymphocytes. Serial proliferation, they analyze how the cells divide, to mean how the cells are dividing or proliferating. And the next is cell variability, to assess how cells are healthy. Cell cycle analysis, they analyze the cell cycle. The rare events, they analyze rare events in the cells. Stem cell analysis, they analyze the stem cells, as you know that they are when they provide the hematopoietic cells which produce the mature cells. Fluorescent protein analysis, they also analyze fluorescent proteins. And here there is the summary of how it works. It works by suspending cell in a fluid stream. You suspend the cell in a fluid stream, then pressing the cell through a laser beam where the light rays are going to press in it. They're detecting the light and scatters is emitted by the cells. Lastly, they analyze the data with a computer. Flow cytometry can be used to analyze samples from many parts of the body, including blood, bone marrow, saliva, and urine. It can also be used to analyze solid tissues such as lymph nodes, the spleen, and mucosal tissues. Mainly, it's used to analyze blood. There is the second question, which asks the principle behind agglutination. Agglutination reactions are based on the specific antigen-antibody interaction. These reactions occur when antibodies bind to particular antigens, such as cells, bacteria, or particles coated with antigens, causing the formation of visible aggregates of clumps. The principle can be summarized as follows. Firstly, they are going to be antigen-antibody binding. Antibodies recognize and bind specifically to antigens on the surface of the cells. Cross-linking. When multiple antigen-antibody complexes form, there is cross-linking which occur to mean they are going to form attraction between them where some bonds are going to hold them together to form antigen-antibody bonds. Clamp formation. Those are the visible clampings which are going to see with the naked eyes after there is agglutination. So in summary, you can say that agglutination principle is antigen-antibody interaction, where antigen and antibody in solutions, they are going to attract each other so that they are going to form bond between them so that we are going to see visible clumping. Types of agglutination. There is dialect where they uses naturally present antigens on the surface. For indirect, they use artificial antigens which are attached to the particles like latex beads which they are made with. Reverse passive agglutination. Antibodies are coated onto particles and the reaction detects specific antigens. Hemagglutination, it's all about red blood cells. Those are agglutination which involve red blood cells, especially in viral detection. The third question is how latex agglutination work in diagnosis. Latex agglutination test. It works by mixing a sample with latex pads that are coated with specific antibody or antigen. To mean that those latex code they are made with certain antibody or antigen according to what it is going to detect. If it's made with antibody, you're going to detect antigen. If it's made with antigen, you're going to detect antibody. So this is how it is done. A sample is sent to a lab and mixed with latex pads. If the suspected substance is present, the latex pads will clump together or agglutinate. The clump become visible to the naked eye. So rats are used to confirm the presence of specific pathogens and are often used as first step in diagnosis. They are simple to perform and can be used to diagnose a variety of conditions, including this is the summary of them. Mostly it's for viral diseases which are tested in immunology. Here there is the fourth question. It talks about the principle or whole precipitation reaction in immunology. The principle of precipitation reaction in immunology is based on the specific binding of antibodies to their antigens. To mean how antibodies are specifically going to bind to certain antigen or specific antigen, as we know that they are specific. This binding forms antigen antibody complexes as well as we have immune complexes where antigen antibody react, which when present in optimal amounts aggregate to form visible precipitates. The principle of precipitation reaction in immunology it relies on the specific interaction between antigen and antibodies to form visible aggregates. When they are present in the correct proportions. These reactions are used in various immunological assays to detect, identify, and quantify antigens and antibodies. Let's go to the last question. The radioimmunodiffusion test is an immunoassay used to detect to measure the concentration of a specific antigen in the sample. To mean if you have a sample with a certain antigen and you want to know the concentration of that antigen in that sample. That's where radioimmunodiffusion test is used. It is based on the principle of antigen-antibody diffusion through a gel matrix, usually agar, and the size of the resulting precipitate ring is proportional to the amount of antigen in the sample. The technique is relatively simple, quantitative, to mean it can quantify, and often used to measure concentration of immunoglobulins or antibodies, proteins, or other antigens. Here there is the steps. To prepare, first step is to prepare antibody containing agar gel. Secondly, you create walls for sample and standards. Thirdly, you add known standards and unknown sample to the walls. 4. Incubate to allow antigen diffusion and precipitation formation. If you incubate, you are just going to allow for it the sample to diffuse and precipitation so that it can be formed. Fifth, to measure the diameter of the precipitation rings and the rest steps you are going to plot the standard curve using known concentration and the rest you determine the antigen concentration in the sample by comparing the ring diameter with standard curve. Okay, thank you for your attention and thank you for being with me. This was my presentation. Thank you so much.
Okay.